DNA replication in prokaryotes. All right, let's get started. All right, so this top strand is our DNA before replication. So this magnet is going to be our origin of replication. And this is where our initiator proteins, or these black magnets, are going to bind to and start unwinding our DNA, which is represented down here in our replication bubble. So real quick, phosphodiester bonds are basically here, the whole way through. Hold together our nucleotides and are represented in this drawing down here. So our top strand, you can see we've got our 3' N, our 5' N, bottom strand, our 3' N, our 5' N. So our bottom strand is going to be our leading strand, and this is going to be where DNA replication is continuous. Our top strand is going to be our lagging strand, and that's where DNA replication is going to be discontinuous, which is going to lead to Okazaki fragments. So moving on, our gyrases are going to form at either end of our replication fork, and that's going to reduce the torsional strain on our DNA. Our helicases are going to start at our 3' Ns, represented by these gray magnets, and they're going to move towards the 5' ends, breaking the hydrogen bonds, allowing the DNA to unwind. So that could be represented down here. As this moves, it's breaking these hydrogen bonds. So then our RNA primase is going to form, and that's going to lay the foundation for our DNA replication, where DNA polymerase 3 is going to start at the 3' end, move to the 5' end, and replicate our DNA. So then, DNA Pol 1, represented by this heart magnet, is going to come through and remove our RNA primase, like that, and then it's going to close the gaps in our lagging strand. And real quick, these original blue strands would be our Okazaki fragments. So now that DNA Pol 1 has made its way through, any of these small gaps are called nicks, and that's where ligase comes in. It's going to come in and seal that gap without adding another nucleotide. So what would happen if DNA Pol 1 was not present in our replication? So that would mean our RNA primase would not be removed, the gaps here would not be closed, and ligase could not do its job. So this would be catastrophic for DNA replication. So that's it. Real quick, references would be the Genetics 311 slides by Dr. Halweg and Dr. Jones, and that is it.